Welcome to Zoology Zone, your go-to place for all things biology. Today, we're going to dive into one of the most intriguing and essential structures in the cell, the plasma membrane. Often called the cell membrane, plasma, or simply the biological membrane, this thin, yet incredibly important structure is found in both cells and plant cells. It's like the cell's very own security system, controlling what gets in and what stays out. Imagine a protective wall that keeps everything in check, but also allows communication and transport between the cell and its surroundings. Even though it's found in all living cells, the plasma membrane has its unique characteristics in plants and animals. It's fascinating how such a simple structure plays such a huge role in the life of a cell. Ready to uncover its mysteries? Let's get started. As we dive deeper into the plasma membrane, it's important to understand its key features and remarkable functions. First and foremost, the plasma membrane is thin, yet elastic, allowing it to flex and adapt to the needs of the cell. This flexibility is crucial because cells constantly face changes in their environment, and the membrane ensures that they can survive these dynamic conditions. One of the most essential characteristics of the plasma membrane is its ability to serve as a selective transport barrier. This means it controls the passage of molecules, ions, and nutrients into and out of the cell. This selective permeability is vital for maintaining homeostasis, enabling the cell to carry out its functions efficiently. Additionally, the plasma membrane plays a pivotal role in generating ion concentration across the membrane. This is crucial for creating membrane potential, which powers processes such as nerve transmission, muscle contraction, and the uptake of nutrients. And, just to highlight, the plasma membrane is also known as the cytoplasmic membrane or cell membrane, as it encapsulates the cell, protecting its contents from external harm while allowing communication and interaction with its environment. Now let's take a look at the chemical composition of the plasma membrane. The plasma membrane is primarily made up of lipids, proteins, and carbohydrates, each playing a vital role in maintaining the membrane's structure and function. Lipids. The most abundant component of the plasma membrane is lipids. The four main types of lipids found in biological membranes are phospholipids, glycolipids, sphingolipids, steroids, with cholesterol being a major example. These lipids work together to create a flexible yet sturdy membrane structure. Proteins. Proteins make up a significant part of the plasma membrane and are essential for many cellular functions. They are classified based on their position in the membrane and their function. Based on position, integral, intrinsic, proteins. These proteins are embedded within the lipid bilayer and often span across the membrane. They help transport substances across the membrane and act as receptors. Peripheral, extrinsic, proteins. These proteins are attached to the outer or inner surface of the membrane and mainly support the membrane structure and help in cell signaling. Based on function, transport proteins help move molecules across the membrane and carriers. Structural proteins. This group includes receptor proteins, enzymatic proteins, and cell recognition proteins. They provide support, help in signal reception, catalyze reactions, and assist in identifying other cells. Carbohydrates. Lastly, we have carbohydrates. These are usually attached to proteins or lipids, forming glycoproteins and glycolipids. They are found on the outer surface of the plasma membrane and play an important role in cell recognition, communication, and immune responses. So, in summary, the plasma membrane structure is a complex combination of lipids, proteins, and carbohydrates, all working together to protect the cell and maintain its internal environment. Structure of the plasma membrane, with diagram explanation. Let's now understand the structure of the plasma membrane in detail, using this diagram. Yellow circle 1. Phospholipid structure. Each phospholipid is made of a hydrophilic, water-loving head, which is polar and faces outward, the watery environments inside and outside the cell. Two hydrophobic, water-fearing tails, which are nonpolar and point inward, away from water. These phospholipids arrange themselves into a bilayer, forming the basic structure of the membrane. Now let's go through each label in the diagram, so you can learn to draw and label this on your own. Phospholipid bilayer. Composed of repeating phospholipids. Hydrophilic heads face the outside, extracellular, and inside, cytoplasmic, environment. Hydrophobic tails face each other inside the membrane. This bilayer forms a semi-permeable barrier, only allowing certain substance pass. Integral protein/protein channel. These proteins span across the membrane. They often form channels or pores to transport large or charged molecules. Help in active or passive transport. Peripheral protein. Found on the inner or outer surface of the membrane. They do not span the bilayer. Involved in signaling, support, or anchoring the cytoskeleton. Cholesterol. Embedded between the fatty acid tails. Helps maintain fluidity and stability of the membrane. Prevents it from becoming too rigid or too flexible. Carbohydrate chains. Shown as branched structures attached to proteins or lipids. These form glycoproteins or glycolipids. Important in cell recognition, communication, and immune response. Glycolipid. A lipid with a carbohydrate chain attached. Found on the outer surface of the membrane. Helps in cell recognition and interaction. Glycoprotein. A protein with a carbohydrate chain attached. Also located on the outer surface. Functions as receptors or cell identifiers. Globular protein/hydrophobic alpha helix chain. These may be enzymes or structural components. Some are embedded partially, others span fully. Often used in signal transduction or catalysis. Let's explore the fluid mosaic model of the cell membrane, a model that explains both the structure and dynamic nature of biological membranes. In 1925, scientists Gorder and Grendler extracted lipids from red blood cells of different mammals. Their experiments led to the conclusion that the cell membrane is made of a lipid bilayer. A few years later, in 1935, Daniel and Davson, also referred to as Daniel and Harvey, suggested that the membrane wasn't just lipids, it also had proteins, which they proposed based on studies of surface tension. Then in 1972, Singer and Nicholson proposed the model we widely accept today, the fluid mosaic model. According to this model, the membrane is composed of a lipid bilayer, which provides a flexible and fluid base. Embedded within this bilayer are protein molecules that appear in a mosaic-like arrangement. These proteins are globular in shape and differ in their positioning. There are two main types of membrane proteins: extrinsic peripheral proteins, attached loosely on the polar surfaces of the lipid bilayer. Integral proteins, embedded within the bilayer. Some of these completely span the membrane and are called transmembrane proteins. Many of these proteins, especially those on the outer surface, have carbohydrate chains attached to them. These are glycoproteins. Similarly, some outer surface lipids also have carbohydrates, and they are called glycolipids. Together, these components not only form a barrier but also help in cell communication, recognition, and transport of materials. The cell membrane is not rigid, it behaves like a fluid structure, and this fluidity is mainly due to the movement of lipid molecules within the lipid bilayer. Most lipid molecules do not move from one layer to the other. When they do, it's called flip-flop or transbilayer movement, and this is a rare event. However, they occur more actively in the smooth endoplasmic reticulum. On the other hand, lateral diffusion is very common. Lipid molecules frequently exchange places with their neighboring molecules within the same layer. In addition to this, lipid molecules also rotate rapidly along their long axis. Their hydrocarbon tails are flexible, allowing bending or flexion, which is more pronounced towards the center of the bilayer, and less so near the polar ends. This constant movement helps maintain membrane flexibility, assists in protein distribution, and enables the self-healing property of the membrane. Cholesterol is essential for modulating the fluidity and stability of the cell membrane. It fits snugly between phospholipids, with its hydroxyl group positioned near the polar heads. This orientation anchors cholesterol in place, limiting the movement of fatty acid tails. At low temperatures, cholesterol prevents phospholipids from packing tightly, avoiding membrane rigidity or solidification. At high temperatures, it restricts excessive movement of phospholipids, preventing the membrane from becoming too fluid or leaky. This dual role maintains optimal fluidity across temperature changes, crucial for proper membrane protein function, signal transduction, and processes. Cholesterol also reduces permeability to small water-soluble substances by filling in gaps between phospholipids, strengthening the barrier. Overall, cholesterol acts like a fluidity buffer and mechanical stabilizer, ensuring the membrane remains flexible, yet resilient. In the heart of every living cell, a silent miracle unfolds. A structure, so delicate, yet so vital, is born not from chaos, but from design. De novo, Latin for from the new, is how membranes come into existence. But they do not rise from nothing. Meaning new membrane components are to pre-existing membranes without disrupting their structure or function. All cellular membranes grow from existing ones, which serve as templates. This ensures continuity and preserves membrane identity. Since membranes are composed of complex lipids and proteins in a specific arrangement, using existing membranes as templates allows the correct insertion and orientation of new components. This method maintains functional consistency during processes like cell growth, division, or organelle formation. In short, membrane expansion is not random, it's a highly regulated, template-driven process to ensure structural integrity and cellular function. Let's summarize the whole membrane is involved in cell recognition and adhesion. First, the plasma membrane recognizes one another and stick together to form tissues. It controls what enters, which is vital for the organization, allowing only cellular molecules to pass through. In summary, this selective permeability is much more than just a cell survival. Finally, second, the membrane is involved in cell recognition and adhesion. By acting as a boundary, the plasma membrane safeguards the internal environment of the cell from harmful substances outside. Third, the membrane plays a key role in cell communication. It contains receptor proteins that detect signals from other cells, allowing the cell to respond appropriately to its environment. Fourth, it facilitates the transport of materials. This includes both passive transport, like diffusion, and active transport that requires energy to move substances across the membrane. Fifth, the plasma membrane helps in maintaining homeostasis. It regulates the internal conditions of the cell, keeping them stable despite changes outside. That wraps up our lecture on the plasma membrane and its essential functions. I hope you found this session informative and helpful. Thank you so much for watching. If you enjoyed this video, please don't forget to like, subscribe, and share it with others who might find it useful. Your support means a lot and helps the channel grow. Stay curious, keep learning, and I'll see you in the next lecture here at Zoology Zone.